Today we will discuss Chapter 11, Transcription of the Genetic Code, the biosynthesis of RNA. In the previous chapter, Chapter 10, we discussed the biosynthesis of DNA, which is the replication. We learn how the replication proceed to make the DNA. And now we will learn how to make the RNA. As we know that the central dogma of molecular biology is that DNA make RNA and RNA make protein. The process of making RNA from DNA is called transcription. And then the process of making the protein from RNA is called translation. So both transcription and translation together, we call it the gene expression. And the transcription is the major control point in expression of gene and the production of a protein. In the use of genetic information, one of the strands of the double-stranded DNA molecule is transcribed into a complementary sequence of RNA, and this is the process of transcription. And the RNA sequence differs from DNA in one respect. The DNA base thymine T is replaced by the RNA base uracil U. Of all the DNA in the cell, only some is transcribed. Transcription produces all the types of RNA, including messenger RNA, transfer RNA, ribosomal RNA, snRNA, miRNA, and the siRNA. And the details of RNA transcription differ somewhat in prokaryotes and the eukaryotes. So for example, for the eukaryotic cell, to activate the transcription, you will require much more different type of enzyme protein to participate to activate the transcription. For example, the chromatin remodeler, transcription activator, because these protein enzyme, they help the cell to remove the nucleosome from the promoter region. Therefore, the RNA polymerase will be able to access to their binding site and activate the transcription. So that's the major difference between the prokaryotic transcription and the eukaryotic transcription. So that we know that the process is much more complicated in eukaryotes, involving a large number of transcription factors. Okay, and in chapter 11, we will go through these different topics, including the overview of transcription and the transcription in prokaryotic cell, the transcription regulation in prokaryotes, transcription in eukaryotes, transcription in regulation in eukaryotes, non-coding RNA, structural motif in DNA binding protein, post-transcriptional RNA modification. So mainly post-transcriptional RNA modification is about RNA process. So that, for example, you add 5' cap, 3' poly-A tail, or the splicing. That's mainly happen in a eukaryotic cell. And also we will discuss the ribozyme. Although there are many differences between the eukaryotic cell and the prokaryotic cell in transcription, there are also some general features which will be shared by both eukaryotes and the prokaryotes. So for example, here is a table to show you the general feature of RNA synthesis. RNA is initially synthesized using a DNA template in the process called transcription, and the enzyme that catalyzes the process is DNA-dependent RNA polymerase. And for the DNA replication, we need the DNA polymerase. For the RNA synthesis, the transcription, we need RNA polymerase. And then we will need for ribonucleotide triphosphate, ATP, GTP, CTP, and UTP. And for the transcription, a primer is not needed to initiate the RNA synthesis. And when we learn the DNA replication, we need the primer to initiate the replication. And then RNA chain grows from the 5' to 3', because the RNA chain grows from 5 to 3. Therefore, the RNA polymerase, they will bind to the template strand and move along it in the 3' to 5' direction. Because as we know, the double-stranded DNA, one is 5' to 3', the other one would be 3' to 5', and that's called the anti-parallel. And then if the template is 3 to 5, then we can synthesize the RNA 5 to 3. Okay, so these are the general features of RNA synthesis that can be shared by both prokaryotes and the eukaryotes. And then the transcription in prokaryotes. So we know the coenzyme and holoenzyme. Holoenzyme, that's the complete enzyme, right? Only the presence of sigma factor will be able to help the RNA polymerase to initiate the transcription. And as soon as the RNA, the transcription initiation started, then the sigma factor will depart from the holoenzyme, and then will leave the coenzyme to continue the transcription elongation until the end of the transcription. Okay, and so basically, as I mentioned earlier, the two DNA strands, one is the template strand, and the other one is called the coding strand. Okay. And here's just the picture to show that normally the top strand is a coding strand, which will be exactly the same sequence as the RNA transcript, except the T is replaced by U, all right? And bottom strand, the 3' to 5', that's a template strand. And polymerase, RNA polymerase, use the template strand to synthesize the new RNA, the messenger RNA transcript. And then this can be translated into the protein. Okay, so this is the basic of the transcription. And in eukaryotic cell, again, so you have DNA. The DNA contains both exon and intron, and they have transcribed the template, and then they give you the pre-messenger RNA. Then they have to go through the some process, okay, to splice the intron and keep the exon, and also put the poly A tail, and also put the 5' the cap at the 5', and so that the cap can lead the messenger RNA to the cytoplasm to find the to go to the ribosome and bind with the ribosome, and then start a translation. That's the RNA processing. So these are the difference between the eukaryotic transcription and the prokaryotic transcription. And then the synthesis is again the same. So basically, the difference is only this hydroxy group and with oxygen or not. So basically, this oxygen, all right, attack the alpha phosphate and remove the pyrophosphate, release the pyrophosphate, and then connect the phosphodiester bond. Okay, and so this is the polymerase, RNA polymerase. They use the SDS page gel to show these are different subunits, okay, from the RNA polymerase. So the coenzyme, we have this alpha 2 beta prime. Holoenzyme, you have to add one extra sigma factor, which is important for the promoter recognition. And then the rho factor, which we will discuss later, because rho factor is relevant to the transcription termination. Okay. So so basically the beta and beta prime subunit is they are the catalytic center responsible for the RNA synthesis. And then the two alpha subunit, okay, they can help to perform the promoter recognition, but it's a further upstream promoter recognition, okay. And the sigma factor basically will recognize it directly to those Tata box, okay. So this is just a basic overview of the transcription. And then for the transcription, okay, the transcription unit is a region from the promoter to the terminator, right? From the promoter to the terminator. And then the promoter basically upstream of the transcription start site. The transcription start site is a positive one. Normally we call it positive one. One nucleotide upstream of the transcription start site is a negative one. And then -10, -35, that's the consensus sequence, right? The promoter region, that's where the RNA polymerase bind, and terminator, that's the place where the RNA polymerase stop transcription and then released from the DNA, from the template, okay? And this is just to show the consensus sequence, all right? -10, -35, -10, -35, these are consensus sequence for the promoter region, and then transcription start site, TSS here. And this just to tell you the RNA polymerase, okay, can bind to the DNA. However, only in the presence of sigma, all right? They will find the promoter region, and then they will just come to the promoter region, all right? And then start forming the open promoter because they start open up double helix, okay? So the transcription, we have 3 different phases: initiation, elongation, and termination. And then for the initiation, so in the presence of sigma factor, okay, when you are the complete enzyme, the holoenzyme, then they come to the promoter and bind to the promoter. Then they open up double helix, and then they start synthesizing a few RNA nucleotide, all right? After like a 10, 12 nucleotide, then the sigma factor will depart from the polymerase, and then they begin the elongation. And finally, when they reach the terminator, then they release the RNA polymerase and also the transcript. So that for the initiation, right, if we look at more carefully, there are 5 different steps, all right, sub-steps. So you have closed complex formation at the promoter region, and then you have the intermediate stage, all right, and then you have open complex formation, all right, and then you have the end of abortive initiation is the promoter escape. That's when the sigma factor depart from the holoenzyme, and then it's enter into the transcription elongation. And this is the experiment to determine the location of the transcription initiation, because they use this the specific chemical, the dimethyl sulfide DMS, all right? Because in the when you are the DMS basically will give the methyl group, okay, to A, all right, to a deoxy, to the nitrogenous A, all right, to give the methyl group. And so based on this particular property, so we can take the DNA strand and mix with RNA polymerase, and then you treat these the complex with the DMS, all right? So in the presence of the polymerase, so they will open up this DNA, the double helix DNA, the promoter region, and as soon as they open up the DMS, will methylate those nitrogenous A here, those A, right? So they methylate this A, and then they remove the polymerase. Once they remove the polymerase, then they will form the double helix again, except those regions they have a methylated A, and so that will become, this will become a they cannot form the hydrogen bond anymore. So if you use the S1 nuclease, they will cleave this region. And so basically in the mixture of all these different digestion, then you produce different size of the fragment, and then you perform the electrophoresis, then you will be able to identify the region where the S1 nucleus cleave, and that will indicate the size of the open the transcription bubble. Okay, and so this example that so the first lane here, all right. So you have the RNA polymerase, all right, and no S1 nucleus. So you see nothing, but if you add the S1 nucleus, you can see from +3 to -9. That's the place where the RNA polymerase open up a double helix to start a transcription. All right, so this is the example to show you the experiment to demonstrate how to determine the transcription bubble in the transcription during the transcription initiation. And then so the initiation elongation. So you recognize the promoter, and then you form this closed promoter complex, and then you open up the promoter complex, and then you start the RNA synthesis, and then after 10, 12 nucleotide, then sigma factor depart from this complex. Then this coenzyme continue the synthesis until the terminator. Okay. Chain elongation. So and then so during the transcription, there's a posing and backtracking. So basically the posing allow the ribosome to keep pace with polymerase. So that's the first step of termination. And backtracking is just to read the correct RNA has been incorporated. That's a backtracking. Okay. And now after the initiation and elongation, the next step is the termination. So termination, basically two types of termination, okay, mechanism. One based on the intrinsic terminator. So basically that will be the sequence feature. So basically for the sequence that you will have the inverted repeat sequence and also the AT reach sequence. So the inverted repeat, basically once you transcribe this region, the messenger, the RNA transcript will form this stem loop plus the poly U, and this is a structural signal to make the RNA polymerase depart from the template, and so this is intrinsic termination, and the other mechanism is the role dependent. So the role factor that's I mentioned earlier, the role dependent termination basically. So as soon as the RNA polymerase start elongation, the role factor bind to the newly synthesized transcript, and then they try to catch the RNA polymerase. So the RNA polymerase continue to synthesize until they reach a point, all right? Reach a point that there's the sequence structural feature that will make a hairpin. So the RNA polymerase will stop at that location, and then the role factor will catch the RNA polymerase and cause the RNA polymerase to dissociate from the template. Okay, that's a role dependent termination. So two different mechanisms. One is the intrinsic termination, the other one is role dependent termination. And the transcription regulation in the prokaryotic cell, okay, there are 7 major strategies. The alternative sigma factor, all right, RNA polymerase switching, anti-termination, enhancer, operon, transcription attenuation, and ribose switch. Okay, so for the alternative sigma factor polymerase switching, anti-termination, that's the change of the transcription machinery. Okay. So for example, the alternative sigma factor. So basically you have different sigma factor, then you can control the transcription. Okay. So for example, in the phage, SPO1 that infect the bacillus, and the host RNA polymerase, okay. This is the host RNA polymerase, and then they have this blue, the blue of sigma subunit, all right? And then they transcribe the origin of the origin of the infected viral DNA. So this is from the host. So they transcribe the origin of the viral gene, right? And then this viral origin translate into the early protein GP28, which is the viral sigma factor. Then after that, they will replace the original host sigma factor with the viral sigma, right? And then they can transcribe the middle gene, and the middle gene give you the two proteins that produce through the middle protein. You have GP33, 34, and this basically can help to transcribe the late gene. So that's the alternative sigma factor to control the transcription, okay? And then the switching. So basically the switching, switch the polymerase. So the early transcription, right? So sigma subunit specificity factor. So you transcribe the early gene, then you form this phage RNA polymerase, and then phage polymerase, and then initiate the class 2 and class 3 gene expression. So this is the example in the phage T7, all right? So you have RNA polymerase switching. You switch the polymerase, and here it's the phage SPO1, SPO1 SPO1, all right? And this is phage T7, and then anti-termination. So anti-termination, basically when the phage, bacteria phage infect the bacteria cell, they will try to terminate, all right? Remember, for example, here, they will use the host polymerase and sigma factor to express the early gene, all right? But the bacteria, they have the mechanism, they try to prevent the transcription, so they have early termination. However, the bacteria phage, they have this anti-termination mechanism to overcome this early termination, so that they can continue to express their transcript for the bacteria phage. And enhancer, basically enhancer is like a DNA sequence, right? That can promote the transcription. Okay, that's a DNA sequence bound by the transcription factor, and then they can help to promote the transcription. That's the enhancer. And so, for example, these are the elements in the bacteria sequence. So they have this up element, and this up element can enhance the RNA transcription. And this up element basically will be recognized by the alpha subunit. Remember, I mentioned the RNA polymerase. You have beta subunit is the beta subunit. They are the catalytic center, and then alpha subunit can help to identify the promoter, which will be the up element. And the sigma factor, basically, they recognize the promoter with -10, -35 region. Okay. Then operon, that's a group of operator, promoter, and structural gene code for a protein, right? The example would be the beta galactidase, the operon. And this is the regulation, negative regulation and positive regulation. So basically, for the positive regulation, if there's no binding of the regulated protein, no transcription. Binding, then you have transcription. For the negative, all right? So for the negative, if a binding, no transcription, and no binding, you have a transcription. Okay, that's the negative versus the positive regulation. So this is in the operator. Okay, the operator. So basically, they have the operator. That's the place where the repressor bind. So when the repressor bind to the operator, they will block the RNA polymerase to come to a promoter. So therefore, there is no transcription. However, if there is an inducer, the inducer will bind to the repressor. Therefore, the repressor will not be able to bind to the operator. and then the polymerase will be able to come to the promoter, initiate the transcription, so you can transcribe the operon. Okay. And also to have for the leg operon. Okay, leg operon basically use a lactose as the carbon source, and the leg operon repressed by the glucose because it's the catabolite repression, right? Because you have this cap binding site, right? In the presence of a glucose, the cyclin AMP will not be produced, and therefore the no cyclin AMP, then the cap will not be able to bind here. So if no cap complex, cyclin AMP cap complex bind to the cap binding site, then the RNA polymerase will be not stable when they bind to the promoter region. Okay, only in the presence of a cap, they can cyclin cap complex, they can stabilize the binding RNA polymerase to initiate the transcription. So it's so that's why it's repressed by the glucose. Or it's like here, cyclin AMP bind to the cap, the complex bind to the cap binding site, then they stabilize the RNA polymerase, and then they can initiate the transcription. And then in the presence of glucose, then the cyclin AMP is not produced, then the cap will not be able to bind, then no polymerase, no transcription. Okay. And then the negative control. So a negative control, basically, for the induction, you need to have a co-inducer, right, to inactivate the repressor so that you can have the transcription. And for repression, you have co-repressor, bind to the repressor and then bind to the operator to inhibit the transcription. The transcription attenuation, that's the control of the trip operon. Okay, that's the transcription attenuation. So that's basically attenuation is here. You have the attenuator, right? So they can control this attenuator. They can control the whole operon. And ribose switch. So that's basically it is a region, the 5' untranslated region of a messenger RNA. So they have an aptamer and also an expression platform. And then the aptamer bind to the ligand, and then expression platform can be a terminator. So so this is ribose switch. And then the transcription in the eukaryotic cell. So we have 3 different types of polymerase. There's a 1, 2, 3, and then RNA polymerase 2 is the one that will synthesize the messenger RNA. And polymerase 1, RNA polymerase 1, synthesize 18S, 5S, 28S ribosomal RNA. Polymerase 3 synthesize this tRNA, right, and 5S ribosomal RNA. Okay, that's 3 different polymerase. And then for the class 2 promoter, that's for the RNA polymerase 2 to synthesize the messenger RNA. So the promoter, you have a co-promoter. So basically attract the general transcription factor and the RNA polymerase 2, right? And proximal promoter can help attract the general transcription factor and also the RNA polymerase, and also some specific specialized transcription factor. And then this is the co-promoter element. So you can see the transcription start site initiator. You have +1 and Tata box. That's for eukaryotic cell. So you need normally you have the Tata box and then the TF2B recognition element, initiator downstream element, right? So these are all required. So basically, for the general transcription factor, that means these six, right? These six are general transcription factor: TF2A, TF2B, TF2D, TF2E, TF2F, and TF2H. So basically, these plus the RNA polymerase 2, right? They will form this pre-initiation complex, right? So of course, they have different steps to form the initiation complex, and then to start the initiation. And here is the order of the binding. So basically, the Tata box, right? In the promoter Tata box. First of all, the TF2D will bind to the Tata box, and then TF2A and B will come to a stabilize, right? So basically, the TF2D contain the Tata box binding protein TBP. They bind to the Tata box, right? TF2D bind to the Tata box. Then TF2A and B will be recruited by the RNA polymerase 2, right? They will bind, all right? And then TF2, A and B, they can stabilize the binding. And then after that, TF2F will come as well. TF2F will be recruited, all right? And then after that, the H and E will be recruited to form the pre-initiation complex, all right? So that's the step. So you have the D and AB and DAB complex, and then recruit F, and then finally you have EH. So that'll be the pre-initiation complex, and then you can start the initiation. Then elongation, of course, is controlled by the post site, where the RNA polymerase will hesitate because they have to check to proofread, and also the anti-termination proceed past the normal termination point. And the gene regulation for the transcription, right? So you can have an enhancer or the silencer. These are the regulatory DNA sequence, right? That where the transcription activator will bind or repressor will bind. Okay, so you have enhancer and the silencer. And looping, basically, DNA looping, they can bring the enhancer, right, into the close complex with the promoter, all right, enhancer or silencer to repress the transcription. And another mechanism would be the chromatin remodeling. So chromatin remodeling can be controlled by the chromatin remodeling complex, which is a protein complex that can remove the nucleosome and to let the polymerase to bind the promoter, initiate the transcription. Or you can have the histone acetylation, all right, to change the nucleosome structure so that the polymerase will be able to bind to the promoter and initiate the transcription. That's a chromatin remodeling through either the chromatin remodeling complex or a histone modifying enzyme. And this is just to show the histone, the modification of the histone and the histone code. All right. So because the histone tail, all right, especially H3 and H4, they have many positive residue like lysine arginine, and those positive residue they can interact with the negative DNA. All right. But if you acetylate those positive charge residue, they neutralize the charge, and so that the nucleosome structure has been changed because it becomes loose, and so the polymerase will be able to bind to their promoter region. Okay, so that's why the histone modification also important for the transcriptional regulation. So the switch SNF, that's like the chromatin remodeling complex, they can nucleosome, right? And had histone acetyltransferase that can acetylate, right? Acetylate the histone, right? So these are the chromatin remodeling mechanism to regulate the gene expression. And for the CREB and the CBP, basically the unphosphorylated CREB does not bind to the CREB binding protein, so no transcription occur. But phosphorylation of the CREB, that's specific transcription factor, then they can bind to the CRE binding site, right? And then they can enhance the transcription. Okay, so that's the transcription. And the non-coding RNA. So basically, the non-coding RNA, so there are two main types. We have microRNA and small interfering RNA. So like this, we have this RNA interfering, all right? So double-stranded. So we have double-stranded RNA from the exogenous sources like a virus or the synthetic RNA created in the lab. Okay, it's right. So this double-stranded RNA and can be cleaved by the dicer enzyme, right? By the dicer to yield the siRNA, all right? And on the other side here, that's a pre-mRNA RNA that's from an endogenous source like a gene or transposon, all right? They can be cleaved by the dicer to yield the mRNA, all right? So we have this siRNA from exogenous source, mRNA from the endogenous source, okay? And then these two mRNA and siRNA are bound by the RISC complex. And then that RISC complex, including the agronaut protein, which is AGO protein, and then the passenger strand of the double-strand RNA, okay, is discarded, and then the RISC recruit. So basically, the RISC they will be recruited to the target specific antisense strand, all right? They discard this passenger strand, and then they will bind to the specific target sequence, and then so they will make sure they will have no translation of this messenger RNA. Okay, so that's the kind of the control of the translation, the expression, by controlling those messenger RNA. And this is just structural motif for the DNA binding protein. So DNA binding domain normally have a domain like either one of these specific features, like helix turn helix, HTH, like this, or the zinc finger, or the leucine zipper, right? So these are three commonly seen structures for the DNA binding domain. And those are DNA binding domain, basically you can find in a specific transcription activator or repressor, because they carry this DNA binding domain so that they can bind to their binding site in the promoter to either promote the transcription expression or to repress the transcription. The transcriptional RNA modification. So basically, let's go to the RNA transcription. Okay, the post-transcriptional modification of messenger RNA. So basically, you have to add 5' cap, all right, and then you have to do the poly-A, tail, and RNA splicing. Okay, so RNA splicing, basically you need the spliceome to remove the intron and then connect those exons. and then 5' cap. All right. So basically, you put the 5' cap in the beginning of the RNA transcript, so that they can cap can bring the RNA transcript to the ribosome, and the 3' and poly A tail can help to protect the RNA transcript. Okay, so the 5' cap involves the recognition and processing of different polyadenylation sites in different messenger RNA as well. So that's this is the splicing. Okay, the splicing process. So the splicing process. All right, so that's the end of the chapter 11.